Dr. David Booth, Associate Professor David Booth, has been at the forefront of discovering MS genes in the world. He's based at Westmead Millennium Institute, and David has recently been awarded MSRA's Most Senior Fellowship Award. So, David, could you start off that story, maybe 10 years ago, and
Yes.
from one to however many now there are, and what that means?
Yeah, look, it's been a fantastic time in genetics just in the last 2 years. Before then, it was not a fantastic time. For a long time, we could only find one gene affecting MS. That gene was very good. It indicated for sure that a particular type of T cell was involved in MS, and it supported the development of therapies like interferon beta and Copaxone. But it was the only gene we could find until 2007. In 2007, as a result of all the great discoveries with the mapping of the human genome, the HapMap, the development of chips which allowed you to genotype all the common variants in an individual at a reasonable price. And as a result of that, we found suddenly a second gene, a third gene, a fourth gene, a fifth gene, and as a result of this big experiment that's just been completed in Cambridge, so now it's up around 49 genes affecting MS. And that's going to dominate research for the next few years, trying to go from those genes to coming up with useful therapies in MS. The big unmet need in MS is treatment for PPMS. So a lot of the genetic work is trying to find out if PPMS has different genes causing it than relapsing remitting. And the answer from the data to date is no, exactly the same. They have, just as all the genes which we've identified as associated with relapsing remitting point to immune misfunction, those same genes are associated with PPMS. So genetically there is no difference that we can detect between PPMS and relapsing remitting. And we know that drugs which modulate the immune system have worked in relapsing remitting, but so far they don't seem to work very well in PPMS. Because we know from the genetics that PPMS is at least started in the same way as relapsing remitting, the susceptibility is due to the same genes, it would suggest that the drugs which work in relapsing remitting have a chance of working in PPMS. The processes should be common. It's difficult to read out if the drug's successful or not because you can't look at reduced lesions, you can't look at reduced relapse rates, the most sensitive markers of success. Because of that, they're underrepresented in clinical trials. And so these great new drugs that are just coming out, there's reason to hope that they will actually also work or could also work in PPMS. When I first came to Australia and started working on MS, the critical funding which allowed us to identify IL-7 receptor as the second gene came from the Trish Research Foundation in combination with MSRA. So that was absolutely vital for us. And I think you find all around the country, it's a really important source of umbrella funding.
On my left, immediate left, is Dr. Fiona Mackay, originally a microbiologist, but now a cell biologist, and I'll let Fiona explain the difference. But Dr. Mackay is part of David Booth's team out at Westmead and is investigating a very specific molecule in the development of progressive MS.
Yeah, thanks, Jeremy. So the gene I work on is called IL-7 receptor, and it's quite the mouthful, so my apologies, don't let that put you off. But David's lab discovered back in the early 2000s that IL-7 receptor was associated with MS, but that's all we knew. We knew it was critical in immune function, but we didn't know how it was that the gene made you more susceptible to getting MS. And so we've done a lot of work on the gene since that time, and we now think we know why it is that it makes you more susceptible to MS. And as a result, we're now looking in the lab at developing a new therapy for MS, and that's been a really exciting thing. In order to explain to you what IL-7 receptor does, I'll just have to tell you a little bit about MS. MS, as you're probably aware, is an autoimmune disease, and in MS, T cells of the immune system get activated inappropriately and they begin to attack the brain and the spinal cord, and we think that's what causes the neurological symptoms in MS. Now, in a healthy immune system, we all have T cells that are capable of attacking our brain and our spinal cord, but there's a really highly specialised subset of other T cells that, and they're called regulatory T cells. Their job is to shut down those autoimmune reactions. And so in a normal healthy immune system, we have this population of regulatory T cells, and what they do is they float around the blood and they look for these autoimmune cells and they shut them down straight away. So regulatory T cells are made in the thymus, and they're actually made by another type of cell called a dendritic cell, which you can see here. That's the big ugly sucker in the middle. And this is where IL-7 receptor comes in. What IL-7 receptor is it's a sensor on the surface of the dendritic cells, and it's the thing that receives the messages from the thymus that tell it to make regulatory T cells. So in a normal immune system, a normal healthy immune system, we get these signals coming in from the thymus saying make regulatory T cells, and those regulatory T cells go out and they suppress autoimmunity in the body. Now, what happens if you have the MS susceptibility version of the IL-7 receptor? Well, a lot of the experiments that we've been working on have focused on this question, and what we found is that people with this version of the gene actually do make functional sensor, so it's sitting there on the surface of the dendritic cell, but they also make a bunch of sensor that actually just gets spat out of the cell, and so it's not attached to the cell, and so the cell's not able to receive those signals to make regulatory T cells to suppress autoimmunity. And we think this is why it is that this gene is associated with MS, because the T cells just aren't getting those signals that they need to be told to become regulatory T cells. So we think this whole process is actually impaired. And I guess the exciting thing about this is that a lot of work around the world is pointing to the fact that regulatory T cells aren't doing their job in MS. And so what we're looking at doing is developing a therapy that can boost up the regulatory T cells for people with MS. So we're doing lots of experiments at the moment that are just looking at how can we make regulatory T cells in a tissue culture plate, essentially. So we're using the knowledge that we have about IL-7 receptor, and we're culturing these two cell types together, and we're working out ways to make regulatory T cells. And the hope is that ultimately we will have regulatory T cells for injection as a therapy. So it's been a really exciting road, and you know, still lots of work to be done. Regulatory T cells are a really hot area in immunology, and a lot of work's been done worldwide. And in fact, just last year, the first injections of regulatory T cells happened in humans. And so, you know, we like to think perhaps 5 to 10 years that it would be a possibility for MS. So.